Hello, are there any questions? Let me share the screen. So today is May 17th, and we're supposed to be well along on Chapter 8, Microbial Genetics. If I remember correctly, we just started that.
We only did once the first slide, the objective on that chapter.
So I went through the first slide?
Yeah.
Okay. I was remembering we just, yep, right there is where I got the two here. All right, so we're going to talk about the structure and function of the genetic material, talking about the flow of genetic information. We'll then move on to talking about DNA replication, RNA transcription, and protein translation. We'll then move into the regulation of bacterial gene expression, meaning what regulates turning genes on and off. And to do that, we'll talk about operons. We'll then move into the topic of mutations. And I think that's mostly in your worksheet. And then talk about genetic transfer recombination. And then at the very end, we'll talk about genes and evolution. So any questions about what we're going to be doing?
No.
Okay. Genetics is the study of what genes are, how they carry information, how that information is expressed, and how genes are replicated. Genes are a segment of DNA that encodes a functional product. This functional product coded by a gene is usually a protein, but it doesn't have to be. It can code only for RNA. Okay, any question about that? Most of the genes code for a protein, but there are some genes, not very many, that code for RNA. And I used to know this, it's about 20 or 30 that code for tRNA, and then a dozen or so that code for RNA. Something like that. Don't quote me on the numbers, I'm just pulling that out of the top of my head. The genome is all of the genetic material in a cell, or we could say in an organism if we're talking about a eukaryote. The study of genomes is called genomics. The genotype are the genes of an organism. The expression of these genes is called the phenotype. For example, an individual who has blue eyes has the genotype lowercase b, lowercase b. For an individual who has brown eyes, they have the, well, the phenotype is brown eyes. The genotype is capital B, lowercase b, or capital B, capital B. The capital B means that the gene is dominant. The lowercase b means that the gene is recessive. And that's why capital B, lowercase b codes for brown eyes. And when I mean brown, I mean brownish. When I mean blue, I mean bluish. There are different shades of blue, bluish, and brownish. I'm just going to word that. But when we're talking about simple genetics, bluish eyes are genotype lowercase b, lowercase b, and then brownish eyes are capital B, lowercase b, or two capital B's. Any question about any of that? All right. And if you have greenish eyes, how are those referred to? We say the genotype is lowercase b, lowercase b. Meaning greenish eyes are in the shade of bluish. The chromosome holds all of the genes together. And in most prokaryotes, there's only one chromosome. As you know, in most eukaryotes, there's multiple chromosomes. And here we see a chromosome of E. coli. A chromosome is the strand of DNA woven with proteins, especially in the eukaryotes that have the DNA wrapped around the histone protein. But prokaryotes have proteins in their chromosome as well. It's just not a histone protein. And all the genes are on the chromosomes. When we're talking about DNA, you should remember that it's a polymer of nucleotides. And there's 4 nucleotides that make up DNA: adenine, thymine, cytosine, and guanine. And I don't care if you learn this name or you call them A, T, C, and G, but you do need to know the 4 nucleotides. And here we're saying a single letter abbreviation for the nucleotides. DNA is a double helix associated with proteins. By double helix, we have 1 strand of DNA and then a second strand of DNA. And the 2 strands are only held together by hydrogen bonds in the bases. You should know that there are 2 hydrogen bonds connecting the A and the T, and 3 hydrogen bonds connecting the C and the G. And A always pairs with T, and T vice versa with A, and C always pairs with G, and G with C. The DNA, the single strand of DNA, is not held together by the nucleotides. It's held together by the backbone of deoxyribose, the sugar, and then the phosphate. So for the single strand of DNA, it goes deoxyribose, phosphate, deoxyribose, phosphate. And this is a covalent bond, a very strong bond, holding the DNA backbone together of the single strand of DNA. Now, the hydrogen bonds are weak, and they break and reform all the time, at least at room temperature, or your body temperature. Sorry, I meant body temperature. If they're so weak, how come the DNA is held together? Because DNA, most of the time, is in your cells, 2 strands held together by hydrogen bonds. But I just mentioned the hydrogen bonds are very weak and they break all the time. So how come the DNA is held together? Nobody wants to guess? It's because there are so many hydrogen bonds holding the DNA together. And when this one breaks, well, the others are holding the DNA together. And then this one reforms, and this one up here might break. You should also know that the strands of DNA are anti-parallel. This strand is facing this way, and this strand is facing that way. Anti-parallel. If you want to go into the chemistry, this is the 3' end of the DNA, and that's the 5' end. It has to do with what carbon, the carbon position in the deoxyribose. So this would be the 3' carbon, and this right there is the 5' carbon, or this right there is the 5' carbon. You don't need to know the chemistry of DNA, but you should know that the strands are anti-parallel. They're running in opposite directions. Any question about any of that? All right, let's talk about the flow of information. Within cells, DNA flows from, excuse me, not DNA, the information in DNA flows from DNA to RNA to protein. And from DNA to RNA is transcription, from RNA to protein is translation. I think we've already talked about this. This is known as the central dogma in biology. and that is the information flows from DNA to RNA to protein. It's kind of a law in biology. Any questions about that? Within your cells, this is the only way information flows. But actually, with some viruses, the information does flow from RNA to DNA, and then from DNA back to RNA to protein. And we'll talk about that much later when we're talking about retroviruses. Now, that was the flow of information within a cell. You can also have the flow of information between generations of cells or from a parent to their offspring. So DNA from a parent cell goes through DNA replication, and then the DNA, one strand will move to one side of the cell, the other strand to the other side of the cell, and then the cell will divide. And then the two daughter cells will each get a copy of the parent DNA. And that we call the flow of information between generations of cells, or if you're talking about parent and offspring, it's the flow of information from the parent to the offspring. And with humans, this is going to be a little bit different because you have two parents, not one, and usually you have one child. Okay? I mean, sometimes you have more than one, but most of the time it's just one. Any question about that? Now, the flow of information can also flow between cells of the same generation. And you'll notice I broke this description because I didn't like the way the picture was going and I added in the DNA. What happens is DNA leaves one cell. So some DNA leaves this cell. And oftentimes this cell will die. And then the DNA comes out of the cell. And then we have naked DNA in the environment. And that naked DNA can be taken up by a cell. The cell that takes up DNA we call a recombinant cell. And that is known as recombination. I just didn't like the fact that it had a parent cell sending DNA to the recombinant cell, because oftentimes the DNA goes out in the environment, the cell dies. And we'll talk a little bit more about that later. The only time that it moves directly from the parent cell to, not a parent cell, a one cell to another cell is when this cell transfers a plasmid to that cell. And we'll talk about that later too. Most of the time the DNA comes out of this cell dies, and then the DNA is taken up by that cell. And then this cell is called the recombinant cell because it gains new DNA and it can express a new gene that came from that DNA. Any question about any of that? I thought we had talked about this earlier. Maybe I was mistaken. And that is the DNA flowing within a cell goes from DNA to RNA to protein. And the language is different from DNA, well, to proteins. It's slightly different to nucleotides too. And the difference is this is DNA nucleotides, that's RNA nucleotides, but it's still nucleotides. And the number of letters in the alphabet, there's 4 nucleotides for DNA. There's also 4 nucleotides for RNA. And then the language for proteins is amino acids. And for most cells, the number of letters in the alphabet of amino acid is 20. Meaning for humans, there are 20 different amino acids that can make up a protein. Now for English, the English language, we have 26 different letters. Any question about any of that? Just a cute information about how the language is translated. In DNA, the language would be, that is a dog. So that's encoded in the DNA. And then in RNA, it'll be the same information, but with a different dialogue, something like, that is a dog. And then in protein, the information will be translated as quello è un cane, meaning the same information, but translated in a different language. If anybody knows what that is, they tell me it's not Spanish, so I'm guessing it's Latin or Italian. Oops, didn't mean to do that. You can tell me if you know. All right, so we've talked a little bit about how DNA flows within a cell, and we'll come back to that. Let's move on to how DNA flows between generations of cells. And this we call the vertical flow of information, because the DNA moves from a parent to the offspring. DNA replication is semi-conservative, meaning when DNA replicates, the two strands of DNA separate, and then one strand is used as a template to build the new strand. And the DNA polymerase just reads this nucleotide here and then inserts the correct nucleotide for the new strand. Any question about that? When DNA is replicating, you can note that right here, we have DNA as a single-stranded molecule. And then there's one other time the DNA can be single-stranded, and that is when a gene is being transcribed. Semi-conservative replication means that the black strands are the original strands of DNA, and then the newly replicated strands will have one original strand, which is black, and then one strand, which is new, and they're both that way. I think I mentioned before that the conservative model would have the replicated strand, two original and two new, and that doesn't happen. It's semi-conservative. What happens, as I stated, is it uses one strand as a template. So the DNA polymerase sees this adenine here, the A, and so it puts on the correct corollary nucleotide, which would be T. And how that's done is you put in a T triphosphate, and then you break off two of the phosphates, and that releases energy, which helps the reaction occur. And then you connect this phosphate to that sugar group with a covalent bond. And then the T will hydrogen bond to the A. Okay, any question about any of that? All right, let's watch a little video on DNA replication. Yep, I could have used that. Didn't need to get a new one. You don't need that other one.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, any questions? All right, so there are two strands of DNA. One will be called the leading strand because the DNA polymerase moves directly down the strand, because DNA polymerase has to work in the 5' to 3' direction. And the lagging strand, the DNA cannot move down the strand as the helicase is opening up the DNA. And so what the DNA polymerase does is it makes the DNA in the reverse direction. And so it makes it in small chunks, which are called Okazaki fragments. So one DNA polymerase binds here and then makes that piece of DNA. Another DNA polymerase will bind here and then make this piece of DNA. And then a third DNA polymerase will bind here and make this Okazaki fragment. Now, this DNA polymerase will only have potentially two breaks in the DNA. Where the DNA first started, there will be a gap between this nucleotide and that nucleotide, because we're going to say that nucleotide was the original nucleotide, and then this one was a newly added nucleotide by this DNA polymerase. And so there will be a gap in the DNA where the DNA is not covalently bound. This nucleotide, well, actually it's this phosphate and this ribose right here. And then when this DNA polymerase ends, unless it ends at the end of the chromosome, but if it ends before the end of the chromosome end, then there will be a break in the DNA here. And how that break is repaired is DNA ligase will anneal that break, which I guess was there, and let's say it's right here, okay? And on this one, it would anneal it to this DNA, which is broken there, and we'll connect to that. To this one here, on the lagging strand, there's going to be many breaks. Wherever there's an Okazaki fragment, there will be one break in the newly replicated DNA on this side. And then there will be another break on the other side of the Okazaki fragment. So each Okazaki fragment will have two potential breaks, and the ligase will anneal those breaks. And so there are three important enzymes I really want you to know in DNA replication. The first is the helicase, which opens up the double-stranded DNA and makes it single-stranded. And then the single-stranded DNA, the DNA polymerase, which is the second enzyme you need to know, can bind to that DNA and then use that single-stranded DNA as a template to make the newly replicated DNA. And then the third enzyme you should know is the ligase, and it anneals the DNA. Wherever there's a break, like right here, the DNA ligase will fix that break and then make it a single strand of DNA, meaning that this will be covalently linked to that DNA there. Any question about any of that? All right. I think that's everything for that slide. And that was a little bit more of the video. It went into it in a different fashion, and there's like RNA primase. I'm not going to ask you to know, remember that enzyme, but I do want you to know these three enzymes. And they're all protein enzymes. You should also know that DNA is made in the 5' to 3' direction. The DNA replication starts by an RNA primer being put in. And I'm not going to ask you what all happens with that RNA primer. It has to be removed and replaced with the DNA nucleotides. The leading strand is synthesized continuously, the lagging strand discontinuously, resulting in Okazaki fragments. Dr. Okazaki is the one who discovered that. That's why it's called Okazaki fragments. And then you should know that DNA ligase anneals the DNA. All right, the synthesis of RNA happens from DNA. DNA is transcribed to make RNA, and the gene can either code for messenger RNA, tRNA, or rRNA. Here's a question for you. How does the RNA polymerase making the RNA know when to make messenger RNA, tRNA, or rRNA? Come on, somebody should guess. It's actually not that difficult.
Based on the genes,
Yeah, it's based on the gene. If the gene codes for messenger RNA or protein, the gene will be transcribed as messenger RNA. But if the gene codes for tRNA, the gene will be transcribed as tRNA. If the gene codes for rRNA, the gene will be transcribed as rRNA. And so the RNA polymerase doesn't do any of it. It's just whatever the gene codes for. Transcription begins when the RNA polymerase binds to DNA. And the RNA polymerase binds at a site that is called the promoter. So the promoter is a region of DNA, meaning certain nucleotides of DNA, that's upstream of a gene. And the promoter, all it does is it codes for the RNA polymerase to bind to the DNA at this site. Any questions about that? Transcription then proceeds in the 5' to 3' direction. So actually, almost everything in DNA happens in the 5' to 3' direction, meaning RNA is made in the 5' to 3' direction. And then transcription stops when the RNA polymerase reaches the terminator. A terminator is a region of DNA after the gene, and what it does is it codes for the RNA polymerase to fall off of the DNA, and that stops transcription. Any question about any of that? So we've discussed two regions of DNA, and they flank a gene. One is a promoter, it's upstream of the gene, and then there's the terminator, it's downstream of the gene. And these regions are important for transcribing the gene. All right, here's a question for you. When DNA replication occurs, normally all of the DNA is replicated. When RNA transcription occurs, only some of the DNA is transcribed. What regions of the DNA is transcribed and what regions are not transcribed?
The protein, where, because RNA synthesize proteins.
RNA polymerase doesn't synthesize protein. Protein gets made in translation.
But does it only transcribe the genes that?
Yeah, that's what I was trying to get at. And that is transcription only happens in the genes. All of the DNA that is not a gene, meaning it doesn't code for a product, that region of the DNA is not transcribed. Any questions about any of that? Now, you might say, well, how much of the DNA is not coding for a gene? And actually in eukaryotes, most of the DNA is not coding for a gene, meaning most of the DNA is not a gene or genes, okay? It's holding the genes together, but it's not a gene. Does that make sense? Now in bacteria, most of the DNA most of the DNA is made up of genes. And we call the DNA that is not a gene non-coding DNA. And then the DNA, which are genes, we call that coding DNA. Any questions about any of that? The important part is that the non-coding DNA is not transcribed. And so for eukaryotes, most of the DNA is not transcribed. Any questions about any of that? All right, we're actually looking at DNA transcription here. There's the RNA polymerase. And I don't know, maybe it's making the RNA right there, and there's the DNA. And that's showing you what's going on in transcription. Remember, this is an oversimplification. DNA can be transcribed as rRNA or tRNA. in which case there's no translation into protein. But most of the genes will be transcribed as messenger RNA, and then the messenger RNA will be translated into protein. All right, let's talk about transcription in a little more detail. I'm only going to talk about this right here, and then I'll move to the next slide. DNA transcription begins when the RNA polymerase binds to the promoter. And when that happens, the DNA is made single-stranded. And then the RNA polymerase can move down the DNA, which is probably shown better in this picture. RNA polymerase then moves down the DNA, transcribing the RNA, which will then be alongside the DNA, and it'll actually hydrogen bond to the DNA. Any questions about any of that? If not, let's watch another little video on RNA transcription. Oh, video is not available. That's interesting. That's new. Let me come see if I have another link. All right, let's look for YouTube. And, oops. See if I can quickly find one. That looks pretty short. I think this is the one we want, from DNA to protein. Maybe not. I'm not sure which one of these I want. So we'll try this one. We're going to start with a commercial.
Grammarly does more than catch errors. With Grammarly, you can find really good, no, perfect words.
In order for the messenger RNA proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. The motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing, and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger.
Okay, any questions about that? All right, so RNA polymerase binds to promoter and then moves down a single strand of DNA, synthesizing the RNA, which we call transcription. When the RNA polymerase reaches the terminator, the RNA polymerase falls off the DNA. That releases the messenger, or the RNA, I should say, the messenger RNA most of the time. And then the DNA will snap back together, meaning a double-stranded DNA. All right, any questions about transcription? If not, let's move on to translation. This is when the RNA is translated into protein, and that happens to messenger RNA. The synthesis by proteins occurs at the ribosomes, and the ribosomes use the information in the messenger RNA, meaning it's the messenger RNA that codes for the information and will code for the amino acids. Messenger RNA is translated in by 3 groups of nucleotides. And so 3 nucleotides code for 1 amino acid. And this we call a codon. Surprisingly, translation of messenger RNA does not start at the beginning of the messenger RNA. The ribosome has to move down the messenger RNA and then will begin translation at the start codon, which is always AUG. So the ribosome will move down the messenger RNA until it reaches AUG. Once it reaches AUG, this codes to begin protein translation here. The start codon also codes for the amino acid methionine. So the start codon is doing 2 things. It's telling the ribosome, begin protein translation here on the messenger RNA. And it's also telling the ribosome, put in the amino acid methionine. So all proteins, whenever they start being made, they always start with the amino acid methionine. And what happens if the protein doesn't want the protein to begin with the amino acid methionine is that methionine will just be cut off the protein and then would have the finished protein product. The ribosome moves down the messenger RNA, adding on more amino acids for each codon until the ribosome reaches the stop codon, and there are 3 of them. You don't need to remember them because there are 3, but you should know that there are 3 stop codons, and when the ribosome reaches a stop codon, it'll stop protein translation. Okay, You should remember AUG because there's only one of them. And it starts the protein translation as well as it codes for the amino acid methionine. Any question about any of that? All right, each messenger RNA codon codes for
Yes,
one.
Sorry, I did have a question.
Okay,
Is it possible to have like several AUG codons in one strand?
it's possible to have more than one AUG because if the protein codes for the amino acid methionine, AUG is the only codon that codes for that methionine. So yes, AUG can be in the messenger RNA in more than one place. Does that answer your question?
Yes. And then I also had another question. For the
Go ahead. We'll live with the dog.
okay. So for when it stops the like the stop codon, would that affect also the length of the strands? Like,
It would
is it
affect the length of the protein because the protein will not get any larger because translation will stop once there's a stop codon.
Oh, okay.
Okay.
Yeah.
All right. So each messenger RNA codon codes for one amino acid. Most organisms contain 20 different amino acids, but there are a little more than 60 codons possible. So what do you do with the extra codons? Since there's 60 possible codons, a little more than 60, and there's only 20 amino acids. What they do is that there's more than one codon that codes for most of the amino acids. There are two, one of them is methionine. There, right there. There's only one codon that codes for methionine. All right, that's AUG. And let me see if I can find the other one. I think it's right here, tryptophan. There's only one codon that codes for tryptophan. You should only know methionine is AUG and the start codon is AUG. And then some of them have more than one codon for each amino acid. Some of them have two. Can anyone see one? Maybe phenylalanine has two. Some of them have four. Serine has four codons coding for that amino acid. And then some of them have six, like leucine has 6 codons coding for that amino acid. This is known as the degeneracy of the code. And that is more than one codon can code for the same amino acid. And we're talking about degenerate in that sense. And that is that there's more than one codon coding for the same amino acid. Any question about any of that? So don't memorize this table. The only thing you might know is that AUG codes for methionine as well as the start codon. I think I will go to the next slide because this one's rather busy. Protein translation happens when the ribosome binds to the messenger RNA, and then the ribosome, and actually there's 2 ribosomal units which bind together on the messenger RNA, and then the ribosome moves down the messenger RNA and doesn't begin protein translation until the ribosome reaches the codon AUG. Now, there's another component that's needed to begin protein translation, and that is the tRNA. The tRNA is important because it brings in the amino acid, and then the tRNA is actually the one reading the messenger RNA, meaning the anticodon binds to the codon. So if the codon is AUG, then the anticodon will be UAC, and this is the corollary, meaning UAC actually will hydrogen bond to AUG. And the ribosome puts it all together. Let me see if I got that shown on the next slide. Yeah, right there. The ribosome puts it all together. And when it's bound to the messenger RNA, it actually has two, I don't know what you want to call it, channels or holes in it. And the tRNA comes in that channel. And then that's how the tRNA will then hydrogen bond to the codon. So UAC will bind hydrogen bond to UAG. So it's actually the tRNA which is reading the messenger RNA. Okay. Any question about any of that? But for protein translation to occur, you need the messenger RNA, you need the ribosome, which you might remember the ribosome is made up of ribosomal RNA and ribosomal protein. So this is both protein and RNA. And then you need the tRNA, and bound to that tRNA will be the amino acid. All right, so the ribosome has two channels, and the first tRNA will come in and bind to the first codon, which will be AUG. And so that'll bring in the amino acid methionine. And then the second tRNA will come in, and it'll see, in this case, the second codon is UUA. And so only the AAU tRNA can come in, meaning the anticodon AAU. And that'll bring in the amino acid leucine. Then the amino acid hooked to the first tRNA is unhooked, and that amino acid is then bound to the second amino acid. And this process right here happens by an enzyme that is actually RNA. It's part of the RNA of the ribosome. And this is the only enzyme we'll ever discuss, which is RNA. Okay, but the amino acid on the first tRNA is removed, and it is bound to the second amino acid, which is still bound to the second tRNA. You don't need to know the names of the site, the P site and the A site. You just know that there are two channels in the RNA, the ribosome. There's actually a third channel which isn't used, and we're not going to discuss it. And I think that's it for this slide. Then once the amino acid is removed from the tRNA, the first tRNA falls out of the ribosome. And then that will be a signal for the ribosome to move down one codon. And when it moves down, this moves to the first channel. And then the third tRNA can come in the second channel in the ribosome. So the first tRNA is released, and then the ribosome moves down one codon on the messenger RNA, and that means this second channel will be open for the next tRNA to bring in its amino acid. Once again, the enzyme removes the amino acid from this tRNA and then combines it to the amino acid over here. So we have a growing polypeptide chain now. And then this tRNA will fall out and the ribosome will move down one codon. And then the next tRNA will come in. And that process continues, growing the polypeptide chain as the ribosome moves down one codon, one at a time, bringing on the new amino acids brought in by the tRNA. And that process continues until the ribosome reaches the stop codon. When the ribosome reaches the stop codon, the ribosome then falls off of the messenger RNA. The last tRNA is released, and then the protein is released from, I don't know what you want to call it, the ribosome and the messenger RNA. Okay? Any question about any of that? That is how protein translation occurs. Are there any questions on protein translation? If not, let me say that there is a complication, and it's shown here, and that is in both prokaryotes and eukaryotes, you can have more than one messenger RNA being made from the same gene. So this is the messenger RNA being made, and here's another one, and here's another one. And you'll notice they're getting smaller. That's because this one has read more of the gene, and it's transcribed more messenger RNA. And this one is just beginning, and so it's a shorter messenger RNA. Okay, and that happens in both prokaryotes and eukaryotes, and that is that the polymerase can make more than one, well, one polymerase can make one messenger RNA, but you can have another polymerase bound to that same gene making more messenger RNAs. Okay. And in prokaryotes, it's even worse. So here we have the DNA, and there's the messenger RNA being made. It's the line, the red line coming out, or maybe it's a blue line. Let me see what that color is. Yeah, it's a blue line, sorry. That's the messenger RNA being made. But this is happening in the cytoplasm of a prokaryote. And so while the messenger RNA is being made, a ribosome can actually bind to the messenger RNA and begin protein translation before the messenger RNA is even completed. And that's a complication we have in prokaryotes, and that is translation can begin before transcription has ended. And then in both prokaryotes and eukaryotes, you can have more than one messenger RNA being made from the same gene at the same time. Any question about any of that? All right. A final complication is only seen in eukaryotes. And in eukaryotes, when the messenger RNA is made, it cannot leave the nuclear membrane. The pore there will not allow the initial RNA transcript to leave because in eukaryotes, the gene, sorry, that's touching my lip. The gene codes for both exons, regions of the gene that code for protein, and then intron, regions of the gene that do not code for protein. And when the messenger RNA is made, or the RNA is made, the initial transcript will have both exons, regions coding for protein, and then regions not coding for proteins, the introns. And these introns have to be spliced out. That means cut off. And then the exons are spliced together, annealed together. And then the final messenger RNA will only have exons. And now this messenger RNA can leave the nucleus and enter the cytoplasm, where the ribosomes can then bind to it and begin protein translation. Any question about any of that? Anyways, that's the final complication that's in eukaryotes, and that is the genes have exons and introns. Exons code for protein of a messenger RNA, and introns do not. And the introns have to be removed before that messenger RNA can leave the nucleus. All right, so we've talked about the structure and function of genetic material. Now let's move into the regulation of bacterial genes, meaning we're going to talk about gene expression, and we have to talk about operons when we're talking about gene expression. One way to regulate the contents that are in a cell is to control gene expression. If you don't need a gene product, why would you bother having the gene turned on? You can turn it off, and then that gene product will not be made. There are other genes which are always expressed at a fixed rate, such as a constitutive enzyme or a constitutive gene. So these genes are always turned on, and they're always expressed at a fixed rate. But other enzymes are not always made, and they're made only as they are needed. And these we call repressible and inducible enzymes, or repressible, inducible, or inducible gene. And these genes are turned on only when they're needed. They're turned off at other times. Any question about any of that? Now I'll ask you, does it make sense to make a gene product if the gene product is not needed? Hello, anyone going to answer? All right, it doesn't make sense for a cell to make a product the cell does not need, because it costs the cell energy to make that product. And so the cell can turn off that gene, and then the product will not be made, and then the cell will not waste any energy making the product. There are other ways to regulate what is in a cell. You can regulate by epigenetic control means. We're not going to talk about that. You're not going to be tested on it. And there are post-transcriptional control, meaning the messenger RNA will be made, but then it is inactivated. So it is post-transcriptional, and then the gene product isn't made. And you can even do things like inactivate the protein. Okay, also post-transcriptional control. We're not going to talk about that, not going to be tested on it. We are going to talk about regulation by pre-transcriptional control means. And there are two ways of doing that, actually three ways if you call it about epigenetic control. There's an inducible operon, and there's a repressible operon. In the inducible operon, the gene can be induced and turned on. In the repressible operon, the gene can be turned off. Okay, we're only going to be talking about inducible operons because we're only going to be talking about the lactose operon, the lactose gene, and it is an inducible gene. Okay. Ah, I got to here. We'll see. I'll leave that here. To talk about gene expression, I need to talk about the operon. The operon is a model of gene expression. A bacterial operon contains the gene of interest. Whoops, went way too far. Trying to blow this up, scroll it down and blow it up. So the gene of interest in our case is the lactose gene. This lactose gene codes for the enzyme lactase, which digests the sugar lactose into, let me see if I can remember, into glucose and fructose. And that's the Z gene in the lactose operon. Okay. And that's the enzyme, it codes for the enzyme lactase. So that's the gene that we're talking about. But in the lac operon, in the lac operon, there are two other genes besides the lactase gene. There's the Y gene and the A gene. In bacteria, the operons may have one gene of interest, which we call a structural gene, but it may have more than one gene of interest. In the case of the lactose operon, there are actually 3 structural genes in the lactose operon, or the lac operon. There's the Z gene, which codes for the enzyme lactase. There's the Y gene. It codes for the protein transporter in the cell membrane, which transports the sugar lactose into the cell. And then there's the A gene, which is involved in the metabolism of the sugar lactose. The A gene is involved in converting, I didn't say that right. This is lactose. It's made into beta galactose and glucose. And the A gene is involved in converting beta galactose into glucose. So all 3 of the structural genes in the lac operon are involved in the metabolism of lactose. And as far as I know, all of the structural genes and all of the operons are related, meaning you have related genes in the operon. And this is only seen in bacteria where they have more than one structural gene in the operon. And they can have, in a bacteria, they may only have one structural gene, but they can have multiple structural genes in the operon. Now in eukaryotes, for each operon, there's only one structural gene. So we have an operon that has the lactase gene in it, and then we have a operon that has the gene for the protein transporter to bring the sugar lactose into the cell. And then we have a gene for A, an operon for A, and that's to convert the sugar beta galactose into the sugar glucose. Okay. And so for eukaryotes, it's a little more complicated in the sense that we have more operons because there's only one structural gene in each operon. And they're also more complicated because there's more regulation of the gene or of the operon. All right. Any question about that? All right, as I mentioned, the lac operon is an inducible operon. It is induced when the sugar lactose is around. Also, in the operon, you have regulatory regions. And these regulatory regions control the expression of the structural genes. Two regulatory regions that we've already talked about, well, at least we talked about the promoter, is one regulatory region of the operon is the promoter, and that's the site where the RNA polymerase binds to DNA. And then another regulatory region that controls the expression of the operon is the O region, which is the operator. And we'll talk more about that. So these control regions control the expression of the structural gene. Any questions about the operon? All right. Let me shrink that down a little bit. So regulation of gene expression can be induction or by repression. The lac operon is inducible. That means that the gene is normally turned off and it is induced to be turned on when the sugar lactose is around. So when you need the gene to be turned on, it'll be turned on, but when you don't need the gene, it is turned off. How it is turned off is there's another regulatory gene, which is the I. It is not a part of the lac operon. In fact, it is its own operon. And this gene controls the expression of the lac operon. The I gene makes a repressor messenger RNA, which will be translated into the repressor protein. The repressor protein binds to the operator. And when it binds to the operator, the RNA polymerase, when it moves down the DNA, stops at the repressor protein and can't go any further. And then that shuts off the lac gene, meaning the structural gene Z. Lactase will not be made when the repressor protein is bound to the operator. So this I gene is a regulatory gene. It makes a repressor protein that binds to the operator, and it shuts down the expression of the lac operon. When we're talking about E. coli, why do you think the lac operon is most of the time turned off, meaning the lac gene or the lac operon is turned off? Why would the cell not need the enzyme lactase? Well, it's pretty easy why the cell wouldn't need the enzyme lactase, and that is the sugar lactose is not around. How often is E. coli going to see the sugar lactose? Where do you see the sugar lactose? Hello, is anyone there? Should we stop this? The
Lactose is in milk.
Yeah, lactose is in dairy products, and E. coli is not going to be seeing dairy products very often unless the E. coli happens to be on a dairy farm. All right. So E. coli isn't going to see the sugar lactose very often. And so E. coli shuts down the lac operon most of the time because it's not going to have the sugar lactose around. And then the E. coli will turn on the sugar lactose, the lac operon to make the enzyme lactase whenever the sugar lactose is around. Whenever lactose is around, allolactose is around. Allolactose is an isomer of the sugar lactose. So one is right-handed and the other is left-handed. I don't remember which is which, but they're isomers of each other. Whenever lactose is around, allolactose is around. Allolactose is the inducer. It will bind to the repressor protein and inactivate it. When the repressor protein is inactivated, it cannot bind to the operator. And then if that operator isn't bound to the DNA polymerase, RNA polymerase will move down the gene and then transcribe the lactase gene. Any question about any of that? Well, what happens to the protein that's already bound to the operator? Any guesses? Two possibilities. Allolactose doesn't actually bind to the protein when it's bound to the DNA, but that binding of the repressor protein is by a stoichiomic reaction. And every once in a while, that protein falls off of the DNA. Now, if there's lots of repressor protein around, another repressor protein or the same one will bind to the operator, and that'll shut down the expression of the gene. But if allolactose is around, then the repressor protein will bind to it once it falls off the DNA, and that will turn on the expression of the gene. And that is why the lac operon is induced. It is induced to be turned on whenever the sugar lactose or allolactose is around. And then when we have the sugar lactose around, we transcribe the messenger RNA. And what do you notice that's unusual about this messenger RNA? You're getting 3 proteins made from this one messenger RNA. That's a little bit unusual. We've never talked about that, but it does happen with bacterial aperons. All right. I think I'll stop here and we will continue here next time. I need to show the vid. All right, any questions before I end this? All right, I will see you in the lab if we have a lab today. I forgot to look to see if we have a lab today. I'm assuming we do. No. All right, I'll see you. Come on.